really dark in here. I didn't realize how dark it was. I didn't turn any lights on. Let me go turn a light on, just in case I need to do hand gesture and share my screen. I'm gonna do Kahoot in just a second. We got 6 people so far. Hopefully get a few more. We'll go ahead and do this the regular way. So I'll pull up the thing so you guys can start joining. Nope. So it's 8 people joined. We got 4 people joined on the Kahoot. We can wait a minute or two, but then I'll probably want to get started so we don't go too late. If you are somebody that doesn't remember how to join, you just have to go to Kahoot app or kahoot.it, and then you can type in the PIN number on the screen. And that'll stay up the whole time. So if you get kicked out or if you want to join later, you can always find the number on the screen. We're about to get started. Probably as many people as we're gonna get. It's okay. Be sure, yes, we are recording, so that's good. All right. Let's go ahead and get started.
Because holy hell, my man needs to buy paper towels. This latest TikTok PR stunt just about sent me over the edge.
All right, so of course it's our lab midterm review. First thing we're gonna start out with is lab safety. I know you guys heard me complaining about how be sure you wear your lab coats, and of course, wear your lab coats. Yes, show me that you understand about the lab safety, even though there's really not gonna be much risk there, but wear them anyways, please. To show me you understand it. So this is just a little review. I think it's like one question, but okay. Be sure to review all lab safety rules. Any of it could be on the test. Honestly, please just wear your lab coats is pretty much where I'm going with this. So where should broken glass be disposed properly? Boy, if only all the questions were this easy. So this is the sharps container, of course. So there's an easy one for us to be aware of. I mean, honestly, the safety stuff, not hard, right? So next we're going to move on to the stains. Before we get started, the basics of what you want to know for the stains. You want to know the steps in order. I don't care about like you put this one on for 1 minute and then this other stain procedure you did for 5 minutes. No, I just want to know, you tell me which stains are used in which order if I were to ask you. I'm not necessarily going to, but I might ask you, what's the primary stain for this procedure? What's the counter stain for this procedure? You know, what was a mordant for this procedure? And so you might want to know those. Basically what each step is and why we're using it for that stain, right? So each stain that is used and its purpose, that's what that's getting at. Like, for example, crystal violet in the gram stain, we use that because the crystal violet gets stuck in that lattice-like, scaffolding-like framework of the peptidoglycan wall, right? So that's why we use crystal violet, and then why we use iodine is to help fix that in place. Why we use alcohols to decolorize and remove the excess that didn't get stuck. And then we use our counter stain, saffronin, because it has a contrasting color. And I will let you guys know, we're gonna see an image of, we saw yesterday, or not yesterday, the day before, whatever it was, an image of a gram negative. And it's pink, right? And I tell you it's red. That saffronin stain is a red stain. It perceives as pink in that microscope, but pink and red, think of them as being the same, okay? You're never gonna have a pink against a red as like your dichotomy. So don't worry about that, okay? It's never gonna be the choices for one stain. In fact, we could go over the 4 main stains, just briefly what they are. It would be that gram stain. And remember the 2 colors for that, it's gonna be pink, red, quote unquote, or purple. Purple is positive, red is negative, right? Then we had the acid fast stain. So that fuchsia color is positive and the blue is negative. Then we had the capsule stain, where you're going to have that purple background all over. And as long as it's a dark background, and you will usually have a reddish pink cell in there surrounded by a halo if it has a capsule, or there will be no halo if there's no capsule. But that background is the dead giveaway for that one, right? And then we have the endospore stain, where you're going to have mostly red cells with some little tealish green blobs. What I say, bubbles, they look kind of like bubbles hidden throughout. So they'll look like they're 2 different kinds of cells or a mixed culture, but they're not. It is a pure culture. Every single slide that you're going to see in the lab exam, now that I have it set up, I can say confidently 100%. Every single slide that you're going to see is a pure culture. So whatever you're looking at, that is 1 cell type. So in that endospore stain, because I'm going to ask you about it and show you an image of it at some point. What was in this? There's something in this spaceship. Yeah, that's what I get for playing. I always have to have a fidget thing. Here's a just do this because it's made for fidgeting. Anyways, the endospore stain. If you see a bunch of red cells and then there's a whole bunch of little, the red cells usually look like lined up like hairs almost. And then the little bluish teal green blobs somewhere in the blue-green area of color. They look like bubbles in there. Those aren't different kinds of cells. Those aren't, it's not contaminated. It's the endospores. So just FYI when you see that. So those are the four main ones that you should be able to recognize by sight based on what I'm, explaining the color to you as. So there's basically two colors you need to be aware of for each of those. Really, even with the capsule stain, you've got the purple background and the red cells. Yeah, I mean, we have the capsule itself, but it'll be a clear or white area. There's no color stain that does that one. It's just a lack of stain. So, because the capsule does not stain. So those are the main colored stains that we did. Be aware of what the wet mount is used for. So what, if I were to ask you a question on the test, what procedure do we use to look at motility of bacteria? The wet mount. You should be able to pick that out of the lineup. So not difficult, just letting you know you need to know it. So yeah, positive and negatives. And what happens if you skip a step? If I ask you, what would a gram-positive organism look like if you skip the alcohol step, for example? Just be able to walk through it in your mind or on paper if you want to use a scratch paper and figure out what color you think it would be if you missed that alcohol step. Whether it's positive or negative, prepare yourself for any scenario. Okay. All right, so let's get into the questions. All right, what is the purpose of heat fixing? Why do we do it? I've asked this question enough. You guys know it's going to be on the lab test too. It's not just on the lecture. We do it so much, I do want you to be sure that you know what it is. All right, so the whole point to heat fixing is to adhere it to the slide and to kill it. Something I do want to be clear about, because some people did miss it on the exam. Just to reiterate, heat fixing is not required to prepare cells for a stain, right? It does help adhere it to the slide, so the rinsing is easier, so it won't rinse off the cells. If you forget to heat fix it, you're more likely to rinse off the cells. That was in the lecture exam. And then the kill it, so that, they're dead there. You're not going to be getting any contamination when you're handling it. But when you think about the capsule stain, with that one, we mixed that little bit of the stain with the bacteria and then just spread it out, dried it, put the other stain on it, and then dumped it, right? There was no heat fixing in that. So you can stain without heat fixing. You're just going to risk rinsing your sample off of the slide potentially. So that's why we have to be gentle. That's why capsule stain has those special jars, the Copeland jars. What is the counter stain used in the gram stain? All right, so your counter stain is the stain that you're going to use at the very end after you've decolorized. So for the gram stain, that's the saffron, and that's what's going to give it the red color. So in that image that we were looking at, it looked like there was like some pinkish cells. There were like purple blotches in there. Generally, that's the pink that you're going to get from your gram negatives. Okay, so it does have like a tinge to it. You can't remove all of the purple. It's just really hard to do without rinsing out your gram positives completely too. So you might have like a dot here or there of the purple in a pure culture, but if it's a pure culture and it's almost all red, and there's only a few purple dots, that's not gram positive just because you see some purple dots. It's gram negative. It's just didn't get rinsed completely. So be aware of that, because if you were to look at a gram negative on a slide, that somebody prepared properly as a gram negative, there might be some purple in there, 1 or 2 here or there, like we saw on the previous image. But if it's majority pink, then that's gram negative. So look at it that way, okay? The gram positives won't be a question. So I can promise you that if it's a gram positive, those suckers are going to look positive, and that's how they should look when we have fresh cultures available to us. So let me go through this. All right. I do have an image of a gram positive later on, and I'll point it out when we see it, so you know the difference. So what color will a gram negative bacteria be after a gram stain? Like I just said. But this is why it was in my spaceship. Oh, no. Yeah, so it's gonna be red. Red's a negative color, right? And I'm not gonna put on the test ever red or pink. Okay, those won't be there. I don't want it to be like that. If you get either one, I mean, that would be make me happy. But red, I like to think of that as a negative color. So we're gonna stick with red because it's easy to pick out that way. Gram negatives, red. Positives, purple, PP. Okay. All right, what is the first stain used in the capsule stain? A worse head. All right. Capsule stain, your first stain that we're going to be using is the Congo red. All right, so it's going to ask you about it here in a moment. But yes, we're going to have that red stain that we put the drop down, and then we added our little touch of bacteria and mixed it in, or dropped bacteria and mixed it in. We used our broths for that one. And then spread it out like a smear. Yeah. So that was the Congo red that we used. And so you might be thinking to yourself, yeah, but you just told us it was, you know, a purple background, Congo red. And like I said in the procedure as well, the reason that the background is purple and not red is because we are adding that Meneville's stain in after. So the Meneville stain is the second stain that will turn that Congo red background into a purple. Proper purple. Alrighty. So this is going to ask you about what type of stain or what types of stain is the Congo red. Which one would it fall under? So I'll tell you that 2 of these are correct. See if you can find the 2. Okay, so it does stain the background, like we were saying, and the stains that stain the background are negative stains. Typically, they repel the actual surface of the cell, negative on the cell, and then the negative stain, you know, they're like and like do not like each other. So they'll repel each other. That's why we get the background stain instead of a cell stain from that. All right, this image you're about to see is a pure culture. What stain is pictured? And this is exactly like what I was describing with you guys. These are all one species. All right, so this is the endospore stain. Most of you guys got this one. It's going to ask about the red structures here in a moment, but those little bluish like bubbles that were all around, those guys were our endospores in this one. Bluish green, yeah. All right, what is the term for the red structures? All right, so the correct answer for this one is going to be the vegetative cells. The active, metabolically active, dividing portion of the cell or cells that aren't making endospores. Those dyes are called vegetative cells. Then we have the endospores themselves. They are stained in the blue-green color. So always in bacteria, you have, you'll have the vegetative, the dividing active, metabolically active ones, and then the dormant ones that are for the hardy, conditions are going to be the endospores. The other two choices, the trophozoites and the cysts, those terms belong to eukaryotes only, right? So the protozoans. I know most of you guys didn't pick those ones, but the trophozoites will be the active, metabolically active, free eating forms of the protozoans, and the cysts will be like the endospores. They'll be hardy and dormant, okay? But for protozoans and protozoans only. All right, which of the following bacteria would be acid-fast positive? Okay, so the acid-fast bacteria are Mycobacterium. So those are the only ones you need to worry about that are acid-fast, and that's pretty much all I have to say about it. Mycobacterium. Okay. All right, what is the primary stain used in the acid-fast stain? Teeth hurt. Like, you guys ever get one of those headaches that like hurts into your teeth? Gosh. That's what I got going on right now. All right, we've got carbofuscin. Most of you guys got this one. Pretty happy about that. Carbofuscin, fuchsia, it's going to make the positive ones be fuchsia, that pinkish color, a bright pinkish red color. The other thing I want to say about this, carbofuscin is lipid soluble, so it binds with the mycolic acid on the mycobacterium. That's a waxy coating that makes them hardy and resistant to things. And but the carbofuscin, since it's lipid soluble, can interact with that wax. All right, in which staining procedure did we use steam? All right, that is good. So this is a question that in a relatively similar form will be on the exam, just so we're aware. The endospore is correct. This is the one where we used that green stain and dripped it on the whole friggin' 10 minutes with that steam going, and it turns the beakers green and it gets on your skin, but it washes off okay in a day. Anyways, that greenish stain, that's the malachite green that gives it that sort of teal color to those endospores that we saw. And that steam helps to force it into the endospores, right? So that's sort of getting into the question that's going to be on the exam. It's going to kind of ask you why we use the steam, okay? So which of the following structures allow for bacterial motility? If you're a motile bacteria, what could you have to help you get around? This might have been, I don't remember. So I can't remember if this is one of those ones, but it might have been. So I have questions on these Kahoots sometimes that I'll say, select all that apply, and then you can only select one. That should be a dead giveaway that only one of them is the choice. So, because that just means I forgot to go back and say that you could select more than one. But I didn't check. I don't remember what it said at the beginning. Flagella, that's the right answer, and that's the only answer, right? They don't have anything else to help them move. Cool. All right, so for the stains, I know I gave a preface before that had quite a lot of information. The ones I'd kind of be aware of, simple stain. I didn't really go over that one, but there's only one color with it, and it's whatever color you stain it. I also want to point out with the simple stain, because you guys are the ones that hear, you get the benefit of hearing this. Simple stains, remember, don't get tripped up. If I tell you, you have a gram-positive bacteria that you stain with a simple stain using methylene blue, what color will it be after you stain it in the simple stain? Blue. It doesn't matter if it's gram positive, gram negative, your grandma, you know, anything. It doesn't matter what it is. Simple stain, whatever dye you are using, that one dye for the simple stain, that's what color it will be. So don't get tripped up if there is something like that that describes a cell that would have this other color in this other stain. We're not doing that. We're doing the simple stain. Okay, so keep your heads on for those. So easy enough. I usually talk about methylene blue for the simple stain, just so that, you know, I know that's mostly what we learned about as well. So no surprise there. The gram stains, remember, the steps for that, it's going to be crystal violet, then the iodine, then the alcohol, and then the safranin. So positive is going to be purple and negative is going to be red. Know what happens if you forget those steps and know what each of those steps does in the procedure. The acid fast stain, remember that's going to be carbonyl fusion. And we do have a decolorizing step in this one. That's going to be the acid alcohol. And I'm going to ask you about the makeup of it, what it's made of, just so you know. It does have acid and alcohol in it. That's all you need to know. And that helps remove any of that carbonyl fusion that didn't stick to the wax, the mycolic acid. If it did stick, then you're acid fast positive and you're going to be fuchsia. If it didn't stick, then the counter stain, which is methylene blue, negatives will be blue. Okay. Capsule stain, that one's the Congo red, mixed with your sample, spread it out, air dry it, add the medieval stain. That's what makes it turn purple, right? That's the second stain added. Primary stain will be Congo red. That's our negative stain, background stain. Anyways, if you have a capsule, it won't stain at all. You're going to do that gentle dunking in the Copland jar, in the DI water, deionized water. And that's, then we're going to, you know, let it dry gently and then look at it under the microscope. Not too many steps there for that one, really just 2 dyes and rinse. But, you know, you get those nice weird looking circles all over, and that purple background is what is going to be the dead giveaway for that, and the pinkish red cells in it. The wet mount, right, I'll say for the capsule, if it has a capsule, you'll have that halo that's white around the cells. If there's any white around any of the cells, you're going to call it positive for capsule. If you look into the slide and all of them are just purple and pink right up next to each other and no white around it, then that is negative for a capsule. All right, wet mount, that's for the motility. You just use some broth and look at it under the microscope. And then we have the endospore stain. So we have a question. I'll get to the endospore stain in a moment, but I do want to come back to our capsule stain just for a second because we have a question. Are Congo and Meneville's both negative stains? And no, only the Congo stain is a negative stain. The Meneville's kind of will stain everything. So it's like a general stain. It stains the background and the cells. So the Meneville's is what's going to give those cells that like pop of that pink color. And then that pink, and I don't know how it mixes with the red to make that purple, but it does. So yeah, but no, it's not technically a negative stain. And I will also, just to be safe and be clear about it, I won't be asking you about medieval stain, simply because that one is one that we didn't go into detail on. We use it, know that we use it, but you don't need to know like the chemistry going on with that one because it's just more complicated than the other stuff we use. Definitely know the Congo though, because it is our negative stain. It stains our background. So definitely know that one. Okay, so endospore stain, right. That's the one we're going to be the 10 minutes over the steam. We have to use the steam to force that malachite green, our primary stain, into the endospores. I know we use the little paper towel on top. The paper towel isn't what's forcing that in though, right? The paper towel is just there to keep it evenly moist, to keep that stain moist and on your sample. That's all the paper towel is there to do. It is not a scientific thing. And you could easily do this procedure without a paper towel there, but we used it, to keep it even and moist. That's it. So the steam is what's forcing that stain in there. The steam is what's doing that. And so those hardy, thick, keratin coated endospores, the steam is going to force that inside of that. And the malachite green doesn't stay well in regular cells, those vegetative cells. So when we rinse it with water, the deionized water in that case is our decolorizer. It's going to remove any of the malachite green from our vegetative cells. And it works quite well. Even though some of us had a lot of junky green on our slides, if you still were able to find your sample, you could still see that your vegetative cells were red. And that's your, if you had endospores, they stood out. So then you can tell whether you had them or not. So it still works quite well, even though it's a messy one, the more effective stains that we use. So, and remember our counter stain with that one is safranin. So in this case, if you're positive, you're going to have endospores, you're going to have those green bubbles or the green little dots in the cells. Sometimes they look like pills. You'll have those green teal colored structures mixed within your red cells. If you're negative, you'll just have red cells, and that's it. Cool. All right. I know a lot of the stuff that we learned was the stain, so that's why I try to go over those the most. Honestly, this whole second part of the semester, we're never using microscopes again. So I might have you do a gram stain just to so you can get it better, because we didn't really get to do them well with good bacteria. So now we have our good bacteria and they're growing well and everything. So we might do them again. I'll let you know about that when we get closer. But other than that, no more microscope. So, but yeah. Okay, so ELISA. This was one we did relatively, recently. I say know this test inside and out. What I'm talking about is, and everything that we went over on that board, how the direct test versus the indirect test, what an antibody is, what an antigen is, how they're used in these tests, and which one's looking for what, and does the patient have this? And if you're looking for antibody, you better have the antigen. And if you're looking for the antigen, you better have the antibody, because you can't look for one without the other, always. So be aware of that. And anything that has to do with either antibody or antigen in the testing is an immune-based test. It's not always ELISA, but it is immune-based. So keep that in mind as well, because, immunofluorescence or your rapid tests for COVID at home, antigen tests, it's immune-based. So yeah, it has to deal with antibody-antigen interactions. Right, let's go. What does the E stand for in ELISA? Right, great. Well, you guys got this one. So this is in fact going to be enzyme. It is enzyme-linked immunosorbent assay. It's not as tricky as it sounds. Enzyme, we know that the enzyme's important in this test, because the enzyme in that last step where we add that antibody linked to the enzyme, that enzyme is what is going to cleave that it's not caloric, colorimetric chromogen. That's another one that was used in our little handouts, right? The color changing substrate in the final step, that enzyme is what's going to be cleaving that to give us the color change. So if that enzyme isn't there, we don't get the color change. And the only way the enzyme is going to be there is if your enzyme-linked antibody stuck at that last step. And it's only going to stick if you are positive. So remember to go through, I guess, like all those individual wells and what is sticking. And then I want you to imagine what would happen if your patient was negative, and see if you can understand how that process would result in no color change. Typically, one of the other questions that I had for the ELISA that we did in our simulation was about, if we're just adding in all this stuff, why isn't it always just color changing? Why didn't all the wells just always color change? Because we're not rinsing anything out. And you're absolutely right. We didn't do any of the rinses. This was just a simulation so that we could see what the color changes would look like. And we took out the rinse step to make it easier to perform it. In reality, yes, you're going to rinse in between every single step. And so that rinse will get rid of anything that did not stick. And that's how it's supposed to work. So that's the ELISA enzyme linked. That antibody is going to be linked to that enzyme that gives you color change. Immunosorbent, immuno is talking about the antibody aspect of it. Sorbent means we're sticking. That's all that means. Like adsorbing to something. Adsorbing, like we talked about with viruses adsorbing onto cells, that just means sticking to something. Assay. So that's what those words are for ELISA, kind of so you understand what that is about. To remind us all. All right, what is an indirect ELISA looking for? All right, so the question is, does the patient have the antibody? This is just like the ones that we did in our simulation, if you can think of it that way. Indirect also, we're looking for the antibody. So has the patient had this infection based on their reaction to it? Have they reacted to the thing? Have they been exposed to it? But we're not looking for the thing. That's why this is indirect. We're looking to see if they ever reacted to the thing. So do they have the antibody against HIV? Okay, so that's what the question would be asking. So if you're looking to see if your patient has the antibody, you want to know if your patient has HIV antibody in them, then you need to have antigen from HIV in order to test this, right, to make it work. So keep that in mind as well. So indirect, does the patient have the antibody? Direct, does the patient have the antigen? Does the patient have this disease? Can we see if they have this disease? It's directly looking at that. Remember, direct is the one that looks like the sandwich. So you're going to have antibody, antigen, antibody, and sandwiches go directly in your mouth. So that's how I remember that one. All right, so for ELISA, the principles of how the assay works, we've kind of just gone over. This could be antigen, antibody, interaction. Remember, an antibody is a protein that your body makes that will bind specifically with some sort of foreign target. That foreign target will be an antigen. Antigen is anything your body can see as foreign. It can have an immune response to. So that's all that is. Indirect immunoassays versus direct. Indirect, we're looking for the antibody. Direct, we're looking for the antigen. And if you are able to like draw out some wells and draw through what happens for each step in each of those, that is going to show great understanding of those assays. How the color change will happen, that's thanks to the presence of that enzyme or the lack of the enzyme. And it's a good idea to know why that enzyme is there, how it got to stay there, and turn it positive, right? And then rapid testing. Rapid testing, I'm just bringing that up just because it does relate directly to these concepts. It's just another immune test. I'm not going to necessarily test you on that in the exam, I'll be clear. But I would be aware of that concept just because it's a useful thing to know when you go into the medical field. Definitely want to be aware of how rapid testing works. Whether that's you're working on humans or animals doesn't matter. There's often rapid testing involved in both situations, whether you're rapid testing for parvo or you're rapid testing for COVID, you know? So it's a good one to be aware of what it is and how it works. It's usually immune-based that way. I'm trying to think if there's something I'm missing here. I'm talking about what are antibodies and antigens. If you can understand those general explanations or like the definitions that are given in the handout associated with ELISA and all that, or that, yeah, the handout, the worksheet that I usually give you guys with the orange box on it and everything. Yes, those have a little more simplified breakdown of that stuff. Definitely be aware of what antibodies and antigens are, know the difference. And if I have like, what is an antibody and a list of choices, then be able to pick the definition that goes with it. Just hint, hint. I know antibody and antigen. All right, next, the DNA stuff. I know we just went over that like today, but we're still going to test over it right now, and we're going to test over it on the exam. So online stuff and the, that means like that study sheet for the DNA stuff and the transformation simulation that we did. So what does PCR do? See who retained this. All right, so yes, PCR is going to amplify DNA. It's literally going to take a strand of DNA and turn it into two in every single step. So each single strand of DNA will turn into two for each round. That means that in every round, you're going to double. So round one, you're going from 1 to 2. Round 2, you're going from 2 to 4, and so on and so forth. Like I said, I'm going to ask you about how many DNA pieces there will be after X number of rounds. Okay, I think I told one class there's going to be 8 or less. I won't ask you about any rounds higher than that for sure. It will actually be less than 8 for sure. But yeah, you can calculate out up to that, then you're definitely golden. Which DNA fragment is the largest from these choices? I swear sometimes that like, if I'm not like the poster child for ADHD, I'm not sure who would be. Always have to have a lot going on. Okay, so which DNA fragment is the largest? So this is going to be, we're definitely looking for KBP here, and there's only 2 of them that have KBP, the 4 KBP and then the 35 KBP. K means 1,000. So you can just say 35,000. The red was 4,000. The blue was 1,200 or 1,200. And then the yellow was 100. So that's the lowest one. So our largest one will be the 35. Don't get tripped up by it just being 35. Keep in mind, you need to pay attention to those Ks that are in there, okay? There is one. Sometimes there are. Which of the following DNA fragments would travel the farthest on an agarose gel? Yes, this question comes up all the time, so it's definitely one that you know is going to be on the exam. Just to point out, these are the same number choices as the last slide. Right. So this is going to be the one that goes the farthest is going to be this smallest one. Smallest is more able to, you can think of it as like a net. Small things can travel through the little spaces of the net. The large ones get caught up in them. So this is going to be 100 base pairs, which is our smallest one. What type of cells are capable of taking up exogenous DNA? Okay, so there's a term related to this, and that is competent. The definition of a competent bacterial cell is one that can take up exogenous DNA. That's what it is. So if it is capable of taking up exogenous DNA, we call it competent. So that's what we learned about in the transformation experiment today, right? Our E. coli cells in that transformation experiment were, our E. coli cells, our Tupperware, were competent cells. So they were able to take up that exogenous plasmid in the mixture surrounding them once we heated them up. And then they held on to it once we cooled them down, right? I would be aware, because I'm pretty sure this is all we're talking about with the DNA stuff. Yes. I would be aware of what happens to your E. coli that you put the plasmid in, your P-glow plasmid, the one that we learned about today. Okay. If you played it on regular, those 3 kinds of media, let's just say that. Okay. All right, so DNA structure and function. I'm not going to ask big details on that. That was what the lecture was for, but I'd be aware of the structure and function of DNA, just so that you're aware of how these things, what the point even is of this stuff, right? Because when we talk about PCR and how it actually works with those 3 steps, denature, anneal, elongate, that it's really important to understand how the denaturing and the annealing and stuff are, and the elongation is happening. Because if you don't understand how PCR operates that way, then you're not understanding PCR really at all. So yeah, so DNA structure and function, general knowledge there. DNA fragment sizes, we know that's based on base pairs. Okay, BP. KBP is kilobase pairs, that's 1,000. Just like a kilometer, that sort of thing, kilograms, that sort of thing. And then we have MBP, which is mega base pairs, which is millions. So know those abbreviations, okay? Agarose gel electrophoresis principles. Again, this was in the lecture, but your DNA has a negative charge because it has phosphates in it. Phosphates we already know is negatively charged. We keep talking about it. So the negative is going to go to the positive end when the electricity is flowing through the gel, right? The gel that we're running the DNA through is called agarose. It's a derivative of agar. It's like the sugar from it, and then they made a gel out of it. It's like more pure. So that's nice. But anyways, that's what it is. And that's how the stuff will get caught up in it. So the larger DNA molecules will get caught up in it like a big old net. And then the small ones can just, squeeze through just fine. They're not blocked by it at all. So they can go way faster. So if it's a race, the smaller ones, think of them as being lighter, they can run a little faster than the heavier ones, right? So that's the idea of how those will work in the agarose gel electrophoresis. PCR, how it works in a practical sense. What I'm saying is those 3 steps. The denature, that's the heat step. We're gonna break those hydrogen bonds between the 2 strands. Anneal means sticking back to it. So we're going to be sticking our primers onto it. So you have to be targeting a gene, specific gene associated with a specific disease or something, right? Specific gene. And then elongation, that's DNA polymerase, that's going to add on nucleotides from there. And so you need nucleotides in your little buffer and whatever else you need to keep the pH normal and everything like that. So that's how PCR would work. And it just repeats that over and over and over and over and over again. In 35 rounds, you'll have like millions of copies. Pretty quick stuff. So it's pretty impressive. Right. Bacterial transformation, steps and results on each type of media. You kind of already talked about that. Steps are easy. So you're just going to mix the plasmid solution with the bacteria, heat it up to open up your bacteria. The plasmid can get in now, go figure. Then you're going to put it onto ice to seal that back in there. And that's it. You've transformed your bacteria. But how do you get your product from it? You're going to plate it on those different types of media. Nutrient agar tells you whether you have viable cells, but it doesn't tell you whether you have the plasmid or not. The nutrient agar with the ampicillin, now only your cells with your plasmid are going to grow, but they're only making the ampicillin resistance product to be resistant to ampicillin. And then if you put it on ampicillin with arabinose media, you're still only growing the bacteria that have the plasmid. So you don't have any extraneous growth going on there. Because if you take away the ampicillin in that point and you just grow, this was a good question somebody had in the lab. You take your transformed bacteria sample and just grow it on arabinose media. What are you going to have? Will you have your product made? Yeah, you'll have your product made by the bacteria that have your gene in it, right? Your plasmid. But you'll also have a whole bunch of growth of bacteria that didn't take it, right? So in order to only grow the bacteria with your target, that's why we're going to grow it on ampicillin with arabinose. Remember, if you are triggering arabinose digestion breakdown, those genes, then you're also triggering making your GMP. They have to go, GFP, they have to go together. So green fluorescent protein will only be made if you are breaking down arabinose. Okay. All right, cool. Plasmids are circular DNA. They're really small in comparison to the chromosome. They are double-stranded though. They have some extra genes on them, but they are not essential to the survival of a bacterium. But they are DNA. All right. Eukaryotes. So I say review your drawings. Isn't that a nice preface to this? Which organism is this one? Because this is really pretty much what the exam will look like with your question. Okay, so this definitely was, and you guys were good for this one, either going to be Penicillium or Aspergillus, because those are the 2 fungi that we really looked at. And remember those structures that we were looking at, like they look like little pink paintbrushes on this one. Those are conidiophores. And remember Aspergillus That one was like that teal green color, and its conidiophores looked like little dandelions like you would blow and the little pieces go in the wind. Yeah, so they're both conidiophores. Aspergillus was the teal green one with the dandelion structures, and the penicillium was the pink paintbrush looking guys. I would definitely focus on those two. Hint, hint. Know the difference between those two? You should be good on that question, okay? And that structure, the structures, remember that word, the conidiophores. It is on your eukaryote study sheet for those two, if you're looking for how to spell it and stuff. What is the name of this organism pictured? I warned some of you guys about this, and I will continue, I will warn you again, but. All right, so this was a picture of Giardia, and that one will not be on the test. There will be an organism that looks similar to Giardia, and it's gonna be that pink color. Our Giardia, just so you know, is like a gray color in the microscopes. This other organism has that similar pink color and looks very similar, but they kind of bunch up more in the smear that's in that. And I think I warned you guys some of the things that I would definitely be aware of. Pink, kind of similar to this image of Giardia. It's going to be on 40x. It's not going to be on 100, right? Because none of our eukaryotes will be on 100. Okay, if it is on 100, it's bacteria. So if it's 40x or less, it is always going to be eukaryotes. This one will be on 40x, and it'll look pink like that, and it'll kind of bunch together. They're called, trichomonas, okay? That's what it's going to be, Trichomonas vaginalis, okay? Be aware of that. One of your choices is going to be Giardia. Do not pick Giardia. It is not Giardia, okay? It is not. Not in the microscope, it won't be, okay? It will be Trichomonas vaginalis, and I will probably bring it up again before the exam just so that, because I don't feel bad if not everybody hears it. Just because I don't know. The eukaryotes, it's just, I hate looking at pre-made slides. I had real samples for you guys to look at. I feel like you'd be learning it. Let me just, I don't know. You know what I'm saying. So anyways, the eukaryotes, your aspergillus versus your, I'm trying to see if I can help, how much I can help you out with this. Aspergillus versus penicillium. Okay, that's one of them. Another station, you will have Trichomonas. Okay. And then the other station, you will have a question about structures. What organism do these structures belong to? Okay, so I would just be aware of, the question really should be very easy if you're just aware of the information that's on that sheet in general, honestly. And that will be it. Yes, penicillium is pink and the aspergillus is a teal green, okay? So be aware of those. Important structural and reproductive differences. Don't overthink these, okay? Know a conidiophore, because I told you that's going to be on there. So know what a conidiophore is, and I would definitely know it versus sporangiophore, which is the covered version, which you didn't see, and you're not gonna see it. So don't pick that. And then maybe the general concepts that are on there. Again, this is going to be more of like a recognize your term sort of thing. Helminth eggs, I'm not going to make you choose between things that aren't obviously different, and I mean obviously different. So if you looked in a microscope and you saw that, you know, helminth eggs in general were round, there's some of them that had like a little different shapes to them, right? Or look like open like casings or something, or have little spikes on the side or whatever it is. But if they in general looked round to you, I'm not going to give you choices that all had round ones, and you have to know which round one it is. I'll probably give you a choice of what eukaryote organism did these eggs come from? And then you will have to pick from obvious choices like Giardia, which is not going to have eggs, right? Those are protozoans. Protozoans don't have eggs. Only the helminths have eggs. Let me reiterate that. Only the helminths have eggs. Only the helminths have eggs, okay? So that's kind of also a little bit of a giveaway there. So the other ones aren't going to have any eggs. Bacteria don't have eggs. Viruses don't have eggs. The protozoans don't even have eggs. Only the helminths do. They're animals. So they kind of fall into that category. So I hope that helps. It should be obvious for that one. And trophozoite versus cyst. Remember that the trophozoite is the protozoan, free living, metabolically active, right? Think of a trough that you eat from. When are you eating? When you're alive and active. If you're dormant, like a cyst, you ain't eating, right? So don't confuse those two. Those are protozoan terms. Identify based on the differences. Like I said, you're going to be looking for kind of obvious differences. And it's going to be as obvious as, if I don't think it's obvious, I tell you the answer, like trichomonas. And if I tell you that you have to know the difference between, and I'm already like stressing over, like, be sure you know the difference between penicillium and aspergillus. And I feel like those are like just hella different from one another when you look at them in the microscope. That's an easy difference. That's the kind of difference I'm trying to get you guys to know. Okay, so don't stress over little tiny differences. Transmission. There is a part of your eukaryote study sheet that will talk about like kind of life cycles and stuff. I think there's one for like Tinea solium. That is tapeworms. So I would kind of just be aware of the general, like how it's transmitted and the basic, how you get it and what form, how does it get into your body and how it gets to, it goes in the gut eventually, right? And goes along there. Well, how does it get there? And what happens after that? So yes. One of the more common, I shouldn't say most common, one of the more common parasitic infections in the United States is neurocysticercosis. And that is whenever you eat tapeworm segment without it being, you usually get it from eating undercooked meat. So if you eat it like just the segments, like people used to say for weight loss or whatever, it's less likely to become a tapeworm in your belly and more likely to become a cyst in your brain. And they encase themselves in this like coating that if you break it open and it oozes out, it causes it to like cause serious, like almost immediate inflammation in the brain that's like irreversible. And you can die very, very quickly if you rupture that. It's almost impossible to like remove these from your brain. So don't ever take a little tapeworm pill. That's the little segments that they have. Don't do it. It's bad. So we do have that in the United States, so be aware of it. Cook your meat. All right, moving on from the eukaryotes. We have the general bacterial growth. So this is just talking about what media is and what how they're growing, colony morphology, culture techniques, that sort of stuff. I think we're going to get into that. So which is not a term used to describe colony morphology? So I would definitely look over your colony morphology terms on that lab 2 sheet and just be able, like if I ask you, if I give you a list of words from science that we've learned, and I say which one is the colony morphology term, be able to pick it out as colony morphology as opposed to like, cell shape or like this one, hyphae. Hyphae is a structure associated with the fungi. So remember the fungi when they have their little like furries that go off, like the hair-like appendages, the growths that come off of their whole little mold body. Those are the hyphae. So that's associated with mold. So it's not colony morphology. Colony morphology is terms to describe the colony on your plate, especially the bacteria, is the ones that we're learning about the most. What is the name of the type of culture that is pictured? There will be some questions on the test that are kind of like this, should be easy. Right, so obviously this is a slant. So they are just like those black top tubes that we've been using, an auger slant. And so they're solid at an angle in a tube. They've got auger in them. So what was this organism? How was this organism isolated? Right, what was the method used to isolate? So this is a streak plate or a quad streak plate, however you want to look at it. Both are correct. So we have those 4 quadrants that we went into, and where you flame between each one to help dilute out the sample each time. So where are you going to see most likely the most individual single colonies? That's going to be in the 4th quadrant, okay? That's why we do it, to get those individual colonies and start a pure culture with an individual colony. Has to be an individual one. So which arrangement of bacteria or arrangements do you observe here? All right, so this is truly strepto. It's not quite as strepto as the sample that I had for you guys that I was showing you on the capsule stand. If you remember, like those pink dots that were all lined up perfectly, those were true streptococci. This is still strepto. We do have that chain arrangement. And the reason, and I know you guys picked the right answer, but the reason it's not going to be cocci or bacillus, I'm not asking about the cell shape, right? That's a cell shape question. This is asking about the arrangement or the grouping. And you guys got it right on the nose. Strepto, that's the chain. If it had been in irregular bunches, kind of Oliver, those would be staphylo, right? Right. Oh, that was like a good example of a gram-positive, by the way. You saw how purple that was? They're like purple, okay? So what's the name of this shape? It's one we didn't focus on. This is a bacterium. What word would you use to describe the shape of these guys? Okay, so this is Spirillum. It's a spiral. It's one that we didn't see on any of our slides because I don't have an example of it. I only have cocci or bacilli, which means the slides that I'm going to be providing you guys to look at for the test is either going to be cocci or bacilli. So that is it, but it's kind of good to see a little bit of variety here. The blue one, the blue choice on here that nobody chose, that umbonate word, that is a colony morphology word. Just FYI, that's one you might want to just be aware, though. Yeah, that's colony morphology word. I don't need you to be able to define it. Just recognize it as colony morphology word and you should be fine. Okay. General bacterial growth, colony morphology, be aware of the terms. Just know that they are terms to describe colony morphology. Media types, broth is liquid. The slant is the slanted auger in the tube. We have the plate auger. That's really all we need. Oh, the butt, that you guys haven't seen it yet, but we're gonna see it. We're gonna see butts later, guys. They're in a tube and they're auger and they're just like, it looks like broth, but it's solid. So they're just like solid in the tube down there, like a broth would be. So that's what a butt looks like. So those are 4 of them that I'd be aware of. Isolation techniques. I say techniques. I'm not gonna ask about any of the others, like diluting or something. Streak plate, okay? Streak plate, know what it's called, know what it's used for, isolation, know which quadrant is most likely to have individual, 4th quadrant, and that sort of stuff. And maybe why we flame it in between the steps, right? To help dilute it out, reset it so we can start diluting out in the next quadrant. All right, cell shapes, cocci and bacilli. Those are the ones you need to know. Cocci are round, bacilli are rod shaped. So how else I want to call it, rod shape. They sometimes look longer, like little hairs or something, but they're rods, they're not round, right? Then we have the cell arrangements. Staphylo, strepto are the big 2 that you would be likely to see on the exam. I don't have examples of anything else, or just no arrangement, but I'm not going to ask you to name that. So, hint, hint. All right, microscopes, how to use them properly. We know number 1, lift your microscope by the arm, right? And put your hand under the base. Remember that your stage is meant to be all the way down and it should be on the 4X when you're putting it away. Then you can put your dust cover over it after it's put away. I have found at least 4 different occasions. I think it's been more than 4, honestly, if I'm being honest. Because I know I pulled out personally 1 microscope from one of you guys' spots. that was on 100 and the stage was all the way up. So I know I said I'd get mad about it. This is your final warning. So I say final warning, we're probably never using the microscopes again this semester. But really, guys, please try to remember that. It's not supposed to play nice with our equipment, okay? But other than that, if you observe a specimen at 40x objective and you have a 10x ocular lens, what is the total magnification? All right, so this is 400x. You just multiply it. So the 40 times the 10, and that's it. Our microscopes have, just to be clear, it's written on the ocular lens, but just to be clear, our ocular lens is 10x, just like all of the examples that I ever give you guys. So if I ask you to take a look at your microscope that you're looking in and tell me what the total magnification is of it as it's set up, as you look through it, you should be able to do this for me. So if it's on the 40x and you're looking at it through the 10x, 400. Serial dilution, we're not gonna need to know it. It's for isolation, but it's literally just diluting something out until it's so diluted that you get individual colonies. So which objective lens do we use for oil immersion? Just so you guys know, I'm not just saying random things. People are asking questions if you guys don't know. So this is the 100x. Most of us are aware of this because we do this. This at least is something that we are familiar with, right? Not too difficult. Remember that oil is for reducing light scatter. Most of you guys know that one. Just a reminder. Calculating magnification, we went over that. Understand the operation. If, and I'm not going to do this on this exam at least. Please don't make me feel like I would need to. I'll say that. But if I were to ask you, and this is probably something I'm going to start instituting in my regular traditional classes, not in summer because I just don't have the time. But if I asked you to, you know, on your own microscope as an individual, have a blank microscope set up 4x stage all the way down, and I give you a slide to bring it into focus on the 100x and ask you to go through the steps in order to do that, and I watched you do it, you should be able to do that correctly without being nervous about you being, doing it correctly or not. Am I doing the right thing or not? You better know how to do the right thing. If you know how to do the right thing, what your parts are named, what those parts do, then you're golden. That's what you need to know for the microscope, right? I think we can all agree, in general, there might be a couple here or there. People come into my class a lot of the time thinking they know how to use a microscope and then quickly learn that they maybe didn't know as well as they thought they did. That's okay, but now you do. So we've learned that, and if I were to ask you to show me, show me and explain to me how it works. Remember the parts, I would say, what is an objective lens? For example, you should be able to tell me what the objective lens was. Point out on the microscope. If I put a piece of tape on an objective lens, you should be able to name it, right? That's what I mean by knowing the parts. And I would say know what their function is, because if I ask you on the test, what is the name of the piece of the microscope, the part of the microscope with the tape on it, and you can tell me that it's the objective lens, and then the next question is, what does it do? Then you'd be able to tell me that it magnifies the specimen. That's simple, but at least know what each part does, okay? And how to use them if I were to ask you to. But like I said, I'm not going to. You don't have to do anything with the microscopes. Just at least if I were to ask you, know it that well, and that's even better. So we were talking about oil immersion. Reduce light scatter, right? So Kirby-Bauer, our antibiotic susceptibility or resistance test. We made our bacteria and stamped them with antibiotics, right? So which antibiotic is this bacterium the least susceptible to? Remember, susceptible is like sensitive. All right, excellent. So you guys pretty much got that. That was E. There was like no zone of inhibition around it. And so yes, so it is not susceptible at all. So it is the least susceptible to that. Don't get too tripped up if I ask you if something is the least susceptible to it. It is not sensitive to it at all, or it is just completely resistant to it. It won't have any zone of inhibition around it at all, like we see with E, if it is like completely resistant, right? So it won't be stopped. It can't be stopped by E. So that was what this question was asking. All right. A zone of inhibition is that clearing area around your antibiotic disc. So know that that's what it's called. That's the zone of inhibition, the zone where growth was inhibited, right? The MIC is the minimum inhibitory concentration, is the concentration, the lowest concentration of that drug that can inhibit the growth. So that's at the edge of the ZOI. So it dilutes out the further out you go. So at that very edge where it's kind of weaker, that is where that minimum inhibitory concentration exists. And there's math to figure it out if you wanted to, but we don't have to worry about it. The Mueller-Hinton auger is the auger that we used for the Kirby-Bauer assay. Why do we use it? Because it has that uniform thickness as well as that pH being controlled, a pH buffer, maintain the pH so that the antibiotic can diffuse evenly throughout the media in that plate and be compared to other plates as well. And then we have bactericidal versus bacteriostatic. I will be asking you about this. If I take a sample, so we have here in our picture, this red bacteria, and we have a zone of inhibition around the K, right? If I take a sample in that clearing, in the zone of inhibition, put it onto some nutrient agar, and then incubate it, and it does not grow, then that means that K is bactericidal to that red bacteria, okay? It killed it. If we take a sample in the zone of inhibition around K, that circle, and we put it on a new plate and it did grow, that would mean that that bacteria is just being inhibited from growing, right? So that is bacteriostatic. It didn't kill it. It's still alive there. We put it on a regular new plate without antibiotic. It grows just fine. It was just like stunted, basically. It was in a stasis, so bacteriostatic. So know that, when we didn't do it hands-on, we did learn about it, and you will have a question about it, okay? About what happens if I take a sample in the zone of inhibition and it doesn't grow, or it does grow. You'll need to tell me what happens, and that's going to be basically telling me, is it bactericidal or bacteriostatic? Now, if you look at a plate like this one in this picture here, and you see that the K has a zone of inhibition around it, can you tell just from that if it is bacteriostatic or bactericidal? If it's killed it, or if it's just, can you tell? No, right? Not just from that plate. What you can tell, the only thing you can tell, is that bacteria couldn't grow there. We don't know why, if it killed it or inhibited it. We don't know the details, but we just know it couldn't grow there. That's all you know. Make sense? I hope. All right, so that's everything for the lab. If you have any questions about any of the material, please contact me. Happy studying. Don't take breaks. Don't crunch too hard tonight, hopefully, or tomorrow morning. Here's our little podiums that I always forget to show. And then I'll post this quiz as well as the recording once it's available. Right. All right, anybody have any other questions? I'm hoping that's pretty good for you guys. So if you guys do have questions, you can always email me. I want everybody to, I don't know how the other day the lab got unlocked before I got down there. I don't really care, but if the lab is unlocked when you guys get down there and I haven't gotten down there yet, do not go in there without me, okay? Just because the exam is set up and it's already got the questions and everything ready. So do not go in there without my permission, because there's rules that you'll have to follow once we get in there and get started. I'll probably post an announcement out about it because I just forgot until just now, but yes, so we will meet outside of that room, probably outside of those friggin' stairs they're going to be doing work on apparently. And hopefully it's not too loud or messy or anything like that, so we'll try to meet right outside of that room. Right before the test should start, and then I'll kind of go over the rules just briefly. And then, we can all just get started, but yeah, we'll meet out there, so. And you can ask any questions if you had any more about this stuff. That would be the time to get it out of your system before we get started, so. All right. All right, that's it, guys. I am gonna go ahead and get off this and load up the exam, whatever this is, the quiz and the video for people. I'll see you guys tomorrow.